Namaskaram to all. Today let's continue the lesson Human Health and Diseases. Now antigens. Antigens are large complex molecular substances that can induce a detectable immune response. The term antigen is used in two senses. The first to describe a molecule which generates an immune response. It is called immunogen. It's a substance capable of initiating an immune response. And the second, a molecule which reacts with antibodies. It is haptens. Haptens are substances that are non-immunogenic but can react with the products of a specific immune response. Next, adjuvants. They are the substances that can enhance the immune response to an antigen. They are called adjuvants. Now epitope. Epitope is an antigenic determinant and is an active part of antigen. Paratope is the antigen binding site. It is the part of an antibody. You can see the picture and can understand. So paratope is a binding site. Okay. Right. Types of antigens. There are two types. Exogenic antigens, endogenic. Exogenous and endogenous. Exogenous. Antigens which enter the host from the outside in the form of microorganisms, pollens, drugs or pollutants are called exogenous. The antigens which are formed within the individual are called endogenous. Example, blood group antigens. Next, antibodies. Antibodies are immunoglobulins, protein molecules synthesized on exposure to antigen that can combine specifically with the antigen. Whenever pathogens enter our body, the B lymphocytes produce an army of proteins called antibodies to fight with them. Thus, they are secreted in response to an antigen by the effect of B cells called plasma cells. The antibodies are classified into 5 major groups. They are immunoglobulin G, immunoglobulin M, immunoglobulin A, immunoglobulin D, immunoglobulin E. Structure of antibody. It is a Y-shaped structure having 4 polypeptide chains, 2 identical light chains, weight 25,000 Dalton, 2 identical heavy chains, weight 50,000 Daltons. Right, both are linked together by disulfide bond. There are 2 terminals, that is regions, C terminal and amino terminal. Okay, so they have variable region at one end and constant region at another end. So variable region only antigens binding site. Okay, functions: agglutination, precipitation, opsonization, neutralization. Agglutination means antibodies goes and capture the antigens, so they agglutinate. Precipitation, அது வீழ்படிவாக்குறது. Then opsonization, அந்த ஆன்டிஜன்ஸ் மேலே அப்படியே முழுகிறது ஆன்டிபாடிஸ், அது ஆப்சனைசேஷன். நியூட்ரலைசேஷன், ஆன்டிஜனை ஃபங்க்ஷன் பண்ண விடாமல் தடுக்கிறது, தட் இஸ் neutralization. Okay, these are the functions of immunoglobulins. Okay, the reaction between an antigen and antibody is the basis for humoral immunity, that is antibody mediated immunity. There are 3 stages. Okay, precipitation, agglutination and finally neutralization. Right, binding force of antigen antibody reaction helps to determine blood groups for transfusion, to study serological ascertainment of exposure to infectious agents, and to determine certain immunodeficiency diseases. Okay, different types of antigen and antibody reactions. The reaction between soluble antigen and antibody leads to visible precipitate formation, which is precipitate reaction. Okay, wow. Whenever a particulate antigen interacts with its antibody, it would result in clumping, I told you, or agglutination of the particulate antigen, which is called agglutination reaction. Right. Next, opsonization or enhanced attachment is the process by which a pathogen is marked of ingestion and destruction by a phagocyte. Opsonization involves the binding of an opsonin. Okay, you can able to see in the picture. Okay, neutralization. reaction or the reaction of antigen antibody that involve the elimination of a harmful effects of bacterial exotoxins or virus by specific antibodies. Okay, next, vaccines. A vaccine is a biological preparation that provides active acquired immunity, okay, to a particular disease and resembles a disease causing microorganisms and is often made from weakened or attenuated or killed forms of microbes. Their toxins are one of its surface proteins. So vaccines teach our body how to defend itself when viruses or bacteria invade it. Okay, so this allows the immune system to recognize the organism without actually experiencing the disease. Some vaccines need to be given more than once, that is booster vaccination, that is vaccination. Okay, right. So there is two different kinds, first generation vaccine and second generation vaccine. First generation vaccine example measles, mumps, rubella, that is MMR vaccine. Second generation vaccine example hepatitis B virus. Okay, first generation vaccine is further subdivided into live attenuated vaccine, the live attenuated vaccine and killed vaccine. Second generation contains pure surface antigen of a pathogen. Right. Vaccination is the process of administering a vaccine into the body or the act of introducing a vaccine into the body to produce immunity to a specific disease, that is called vaccination. So what is immunization? It is the process of the body building up immunity to a particular disease. Okay, so vaccines work by fighting the pathogen and recording it in their memory system. So our immune system having memory, right? Hypersensitivity, that is overactive immune response. The exaggerated response of the immune system to certain antigens present in the environment is called allergy. The substance to which such an immune response is produced are called allergens. Okay, allergy is a form of overactive immune response, as I told you already. It is mediated by IgE, that is immunoglobulin E epsilon and mast cells. Okay, anaphylaxis, very important to mark. It is the classical immediate hypersensitivity reaction. It is a sudden, systematic, severe and immediate hypersensitivity reaction occurring as a result of rapid generalized mast cell degranulation. I have told you many times about AIDS, so I skip HIV. Next, autoimmune disease. Autoimmunity is due to an abnormal immune response in which the immune system fails to properly distinguish between self and non-self and attacks its own body. ஸோ நம்மளோட பாடி செல்ஸை நம்மளோட ஆன்டிபாடிஸை ஆர் நம்மளோட WBCஸை கில் பண்ணுறது ஆர் டெஸ்ட்ராய் பண்ணுறது, தட் இஸ் கால் ஆட்டோ இம்யூன் டிசீஸ். Example, Hashimoto's thyroiditis, Graves' disease, Addison disease. Tumor immunology. A tumor or neoplasm is a group of cells whose growth has gone unchecked. When a tumor continues to grow and invades healthy tissue, it is called cancer. They spread to other parts of the body from the tumor and give rise to secondary tumor. This is known as metastasis. Tumor may be benign or malignant. Benign non-cancerous and malignant is very harmful.